Hello friends, welcome back to BioOne Education. Today's topic powers almost all life on earth. Photosynthesis. We will cover light reaction, the Calvin cycle, C3 versus C4 plants and finish with the exam trick. Let's get straight into it. This chapter scores 3 to 4 questions most years. The whole topic split cleanly into two halves. Light reactions happening in the thylakoid membrane which need the light and produce ATP and NADPH, and the Calvin cycle happening in the stroma which uses that ATP and NADPH to fix carbon dioxide into sugar. Most questions simply test which even happen where. Keep that split in your head and this chapter becomes manageable. Chloroplast structure. Quick recap from episode 1. Photosynthesis happen inside the chloroplast. So let's place our two reactions correctly. The chloroplast has an outer and inner membrane enclosing it, much like mitochondria. Inside flattened sacs called thylakoid stack up into piles called grana. This is exactly where the light reaction happens because that's where chlorophyll sits. Surrounding the grana is fluid matrix called stroma. This is where the Calvin cycle, the dark reaction, takes place. The chloroplast also carries its own DNA and 70S ribosomes, which is strong evidence that it evolved from free-living bacterium through endosymbiosis billions of years ago. Pigments are what capture sunlight. Chlorophyll A is the primary pigment that directly takes the part in the light reaction. Chlorophyll B is an accessory pigment. It captures energy and hands it to chlorophyll A, but cannot work alone. Carotenoids, including carotene and xanthophylls, absorbs extra wavelength and protect chlorophyll from the damage. And remember, leaves look green because chlorophyll reflects green light and absorbs red and blue best. This is why the action spectrum of photosynthesis peaks in at red and blue, not green. Light reaction, the photophoto. Light reaction happen in the thylakoid membrane. Light energy exit electrons from at photosystem 2, then photosystem 1. Water is split at photosystem 2 in what is called photolysis or yield reaction. This releases oxygen and replaces the electron that left electron then flow through the electron transport chain, pumping hydrogen ion into the thylakoid lumen on the way. The two products that come out of this entire light reaction are ATP made by chemiosmosis and NADPH made at photosystem 1. Both of these feed directly into the Calvin cycle next. Here is the light reaction animated. Watch the electron travel from photosystem 2 through the chain to photosystem 1. Hydrogen ions build up in the lumen and the ATP synthase turbine spin to make ATP. This single loop is the entire light reaction. Narrate it as photosystem 2 electron transport chain, photosystem 1 ATP synthase, and you have the whole Calvin cycle happens in the stroma. It uses ATP and NADPH made by the light reaction to fix the carbon dioxide into sugar. Melvin Kelvin discovered this and won a Nobel Prize for it. It is called C3 pathway because the first stable product has 3 carbon. Three steps are three steps. First is carbon fixation. The enzyme Rubisco attaches CO2 and to form ribulose bisphosphate, forming an unstable 6-carbon compound that immediately split into two molecules of 3-phosphoglyceric acid. Reduction. Second is reduction. 3-phosphoglyceric acid converted to glycerol 3-phosphate using ATP and NADPH from the light reaction. Third is regeneration. Most of the glycerol 3 phosphate is recycled to generate ribulose bisphosphate, while one out of every six glycerol 3 phosphate molecule exit the cycle to eventually form glucose. Here is the Calvin cycle as the spinning loop. The orange marker travels around fixation, reduction, regeneration again and again, just like it happened in a real chloroplast, which it loops few times and the three-step order will lock in permanently. How it works: CO2 is the first captured in mesophyll cells by the enzyme phosphoenol pyruvate carboxylase, forming a 4-carbon compound called oxaloacetate. This compound travels into bundle sheath cells which are wrapped tightly around the vein in the pattern called Kranz autonomy, and there it releases CO2 again for the Calvin cycle. The advantage: PEP carboxylase. PEP carboxylase has no affinity for oxygen unlike Rubisco, so there is no photorespiration at the step. And by concentrating CO2 in the bundle sheath, Rubisco there works efficiently without being distracted by oxygen. C3 versus C4 plants. Here is C3 versus C4 in one table, your revision snapshot. In C3 plants, the first CO2 acceptor is ribulose bisphosphate, a 5-carbon molecule, and the first stable product is 3-phosphoglyceric acid, a 3-carbon compound made by Rubisco. There is no Kranz autonomy and photorespiration is significant. Wheat, rice and rose are classic C3 example. In C4 plants, the first acceptor is phosphoenol pyruvate, a 3-carbon molecule forming oxaloacetate, a 4-carbon compound made by phosphoenol pyruvate carboxylase. They have Kranz autonomy and photorespiration is minimal or absent. Maize and sugarcane are classic C4 examples. Screenshot this table. It answers most comparison questions instantly. Photorespiration is the wasteful twin of normal photosynthesis. Rubisco has affinity for both CO2 and oxygen. When it accidentally binds oxygen instead of carbon dioxide, the process makes no ATP, no sugar, and actually releases CO2, pure waste. This happens more in heat, dry, bright conditions when stomata close to save water and oxygen builds up inside the leaf relative to CO2. C4 plants largely dodge the problem because phosphoenol pyruvate carboxylase concentrates CO2 in the bundle sheath cells, so Rubisco rarely encounters oxygen there. There is precisely why C4 crops like maize outperform C3 crops like wheat in hot tropical climate. What controls the rate of photosynthesis? Several factors control how fast photosynthesis runs, and this is governed by Blackman's law of limiting factors. The rate is limited by whichever factor is in shortest supply. Light intensity. Rate rises with more light, then plateaus once light is no longer the bottleneck. CO2 concentration rises with CO2 up to about 0.05%, then plateaus. Temperature. The optimum is around 25 to 35 degrees Celsius. Enzymes like Rubisco slow down or denature outside the range. Water is efficient is indirect since water stress closes tomato and blocks CO2 from entering. Quick recall toolkit. Time together every trick in one card. Light reaction happen in the thylakoid. The Calvin cycle, the dark reaction happens in stomata. C3 plants uses rubisco to make 3-PGA, a 3-carbon product. C4 plant uses PP carboxylase to make oxaloacetate, a 4-carbon product. Kranz autonomy is unique to C4 plants only, and chlorophyll absorbs red and blue light, reflect green. That's why leaves look green. Let's step outside the textbook. Scientists are building artificial leaves, devices that mimic photosystem to split water using sunlight and produce clean nitrogen fuel. Separately, a major global research product is trying to engineer C4 machinery into rice, which is naturally a C3 plant, to boost yield and cut photorespiration losses. Since rice feeds off the plant, on a planetary scale, photosynthesis removes roughly 120 billion tons of carbon dioxide from the atmosphere every year, making plants Earth's single biggest climate regulator. And here is an irony with remembering rubisco is the most abundant enzyme on the planet. Yet it is famously insufficient because it keeps grabbing oxygen by mistake. Fixing that one flaw is one of the biggest target in craft science today. Surprise round. NCERT hidden lines that become question. The oxygen released in photosynthesis comes from water, not carbon dioxide. A very common trick question. Melvin Calvin Melvin Calvin discovered the Calvin cycle. Chlorophyll A is the primary pigment. ATP is made by chemiosmosis at ATP synthase. The first C4 acceptor is PEP. The enzyme is PEP carboxylase. Rubisco has affinity for both CO2 and oxygen, and photorespiration produces no ATP and no sugar. One more favorite, photosystem 1 was actually discovered before photosystem 2, but in the actual light reaction, photosystem 2 acts first. That naming mismatch trips up many students. NEET asked this chapter in 4 costumes: assertion, reason, like C4 plants being more efficient in heat because PEP carboxylase avoids photorespiration. Match the column pairing enzyme, pigment and processes. How many are correct? Mix up C3 and C4 features and diagram based labeling, electron transport chain, or identifying Kranz autonomy. Rapid fire. Pause. Answer in your head. Then play again. 4 or more right, your exam ready for this chapter. Let's wrap up in one breath. The light reaction in the thylakoid makes ATP and NADPH. The Calvin cycle in the stroma uses them to fix CO2 into sugar. And C4 plants add a phosphoenol pyruvate carboxylase step to dodge phosphorespiration in hot climate. For every part, ask where it happened and which enzyme drives it. Final challenge: which enzyme fixes CO2 in C4 mesophile cells? Drop your answer in a comment.